Well, good afternoon. Hello and welcome to this lunch hour lecture. I'm very pleased to introduce Professor Joyce Harper from the Institute for Women's Health here at UCL with her lecture entitled Reproduction Without Sex, What Does Technology Have to Offer? Over to you.
Thank you.
Joyce.
I'm going to come stand over here. Just to be alternative. Right, so it's a great pleasure to give this lunch hour lecture, and as we heard, my title is Reproduction Without Sex, What Does Technology Have to Offer? So it was exactly 5 years ago today, March the 8th, it was International Women's Day when I gave a talk in 2011 on genetic testing in the 21st century. And I was amazed to see that my talk then has got over 15,000 views, so I feel very, very happy that obviously lots of people have watched it. So it's great to be able to give a talk today, 5 years later, on International Women's Day. So what am I going to talk about today? Well, I'm going to talk about in vitro fertilization and just tell you a few of the stories that we have with the technology and where it's going to take us. And I'm going to give you a little update on reproductive genetics. My last talk I gave 5 years ago really concentrated on something we call pre-implantation genetic diagnosis. And I'm going to really take that forward now and see where we've progressed with that in the last 5 years. Now, I started working in IVF in 1987 after I finished my PhD. And technology over that time has really moved on. And I've spent a lot of the time looking at the laboratory aspects of that. And I've written a few papers, this one about when and how should new technology be introduced into the IVF laboratory, and one on the clinical benefit and safety of these procedures. So this is something I've really thought very much about since the time I've been working in this area. But I'm going to really concentrate now on some of the really controversial uses of IVF and of reproductive genetics. So it was in 1978 when everything started with the birth of Louise Brown, and it was Robert Edwards and Robert Steptoe that really did the pioneering work on this in the UK. And this is Bob Edwards here. He won the Nobel Prize in 2010. He was a very good friend of mine. And this is Louise Brown with her child, which she had a few years ago. And I just wanted to ask everyone here, who knows someone who's either been through IVF, you may have been through IVF yourself, or you may be an IVF child. So we've had a few IVF children, it's made me feel very old. A few hands are going up. I'm sure, good, excellent. Okay, I'm sure a lot of you, it affects at least 1 in 10 couples. And as we're going to see through my talk, we're going to actually make things worse in the next few years for various reasons. So just to start so you understand a little bit about IVF, first, what we do in IVF is we give the woman some fertility drugs. It's called superovulation. And we want her to produce more than 1 egg. In a normal menstrual cycle, she would just normally produce 1 egg. In IVF, we want about 8 to 12 eggs so that we can make some embryos and select some good quality embryos to transfer back, which will give us a higher success rate. So this is a human oocyte here. It's the actual oocyte is this part here, and these cells are called cumulus cells, which surround and protect and nurture the early egg. And this is some sperm. We're going to prepare some sperm from the partner, and then we're going to mix them together, and we're going to monitor the embryo, normally up for about 5 days in our IVF laboratory. And you'll see here this, the embryos are surrounded by this thick membrane called the zona pellucida, and this surrounds the egg, and it will stay surrounding the embryo until just before implantation. So we all started off like this. This is our, what we call a zygote. We have the male and female pronuclei here, and these will join together, and then the embryo will start to divide. And on day 2, we'll have around 2 cells, 3 cells, and 4 cells. And day 3, we'll have 8 cells, and 5 and 6 cells. And what we normally do in IVF is we take the dish out of the incubator that's holding these embryos. They're not really grown in test tubes anymore. They were years ago. But they're grown in culture dishes. And the embryologist will have a look at these and monitor how they grow. And I'm just going to show you one of the new technologies. It's called time-lapse. So we can now have the embryos in an incubator that contains a video recorder, and it will actually video those very early stages of development. So instead of just having the static picture that we used to have in IVF, where we just look at the embryo once a day and see what happened, we couldn't tell what was happening in between, and now we have this great time lapse which can show us a continuous video of how early development is happening. So what I'm going to talk about is some of the things that have happened in the last 38 years since Louise Brown was born. How has IVF treatment changed? And this is just the tip of the iceberg. I've only got about half an hour, so I've just picked some things that will give us a nice story about where we are and what technology has to offer to have our reproduction without sex. Well, I'm going to tell you about freezing eggs and where we are with that. I'm going to talk to you about egg and sperm donation and some of the controversial uses of that now. Surrogacy as well, very controversial use of IVF technology. Then I'm going to finish talking about genetic testing with an update of pre-implantation genetic diagnosis. Now first, something that's really happened in the last 5 or 6 years is cross-border reproductive care. Well, actually, it's been happening a lot longer than that, but we've called it very recently cross-border reproductive care. And one of my colleagues, Francoise Shenfield, who works at UCH, she's been leading a lot of work on this with a group that we do a lot of work with called ESHRE. They're called the European Society of Human Reproduction and Embryology. And ESHRE and Francoise's team have published some papers on this, and they've tried to see how much patients go overseas to have fertility treatment. And it used to be called reproductive tourism, but we felt that tourism wasn't a good description of what was actually happening. And so nowadays, when we want to find out information about things, it might be something we want to learn or some medical treatment we might want to have, we're using Google more and more. And you can even use Google to find your fertility treatment. So why would you want to do that? Why wouldn't you just go to the clinic and your doctor and have fertility treatment there? Well, there are rules and regulations in different countries, and it's totally different in different countries, even within Europe, but certainly globally. People have different rules and regulations. So there's some things that will be allowed in the UK that aren't allowed in other countries, and vice versa. It could also be cheaper to have fertility treatment in another country, and it could also be that we have a waiting list in the country you're living in, but you won't if you go to another country. So you can sit and Google. You could go to the Barbados Fertility Clinic, a vacation that really delivers. I think it's a terrible pun. Now, we used to say sun, sea, and sex, but this is to talk about reproduction without sex. And indeed, it's sun, sea, sand, and fertility treatments. And this is just one of many, and this is an area that we're working on at the moment, how we buy fertility treatment on the internet and what that means to a patient. You can even win an IVF holiday in Greece. So this goes on and on. There's so much information about this, and I'm working with the Professor of Law at the London School of Economics, Emily Jackson, and we're looking at how this actually is available to any patients just sitting in their front room and looking at what's available. I'm going to bring this up when we talk about the 4 areas of my talk. So let's first talk about egg freezing. So unfortunately, International Women's Day, we're not a good design. Our eggs are reducing in number and quality daily. So I always say to my students, as they sit here in the lecture rooms, they're becoming more and more infertile with every minute that ticks by, which is very depressing. In utero, we have about 7 million eggs. By birth, we've got about 1 to 2 million eggs, and our golden age that we talk about is about 35 years. The graphs, you can look at numerous graphs of fertility treatment and fertility rates, and at 35 years, fertility really starts to decrease significantly. And over 42, it's really difficult to get pregnant. Over 45, really almost impossible. So female fertility is closely related to the age of the egg. So if we want to get pregnant, we really have to do this when we're younger. We're going to have much more chance of getting pregnant when we're younger. But we've had a look at this globally, and all cultures have increasing age at first conception. So about 20 years ago in the UK, it would have been around mid-20s that most women were having their first baby, and now it's over 30 years of age. But this is true of every country, not just the UK. And so evolution has not kept up with feminism. We cannot we're delaying our fertility, but our fertility is decreasing. And I'm part of the British Fertility Society Fertility Health Task Force, and we're having a meeting next month at the Royal College of Obs and Gynae to discuss how we can get education about fertility into the national curriculum in the UK. The national curriculum talks a lot about sex. It doesn't talk about anything about fertility. And we have to make young people aware that with the lifestyles we're leading now, people are going to delay their fertility, but they will have a problem if it continues to creep up and up past 30, 31 years. So why are women delaying having children? Well, there are many reasons. I've just listed some of them here. They may not have found the right partner, and they may not have found the right partner because they're busy doing other things. They're busy having an education, developing their career, they're travelling, they're enjoying their social life, and I put financial at the end. I think that's one of the least reasons why people don't have children. But women around the world are doing other exciting things. Rather than wanting to settle down and have children in their 20s, as they used to, they're going and doing these things where 50 years ago, probably our mothers were not able, certainly my mother wasn't able to do hardly any of these things. She didn't have the freedom to do that. We do have the freedom, but the problem is our fertility. Women come to our clinics, the average age of women in our IVF clinics is around 38 years of age, so by then their fertility is really affected. If they tried to get pregnant when they were 28, they would have had a much higher chance of getting pregnant than if they leave it till 38. So for some of the patients that come to our clinic, because of their age-related infertility, they're not going to get pregnant. So the options for them are to have egg donation, they could have no children, or they could go for adoption. But now we've got social egg freezing. So there's a technique, a laboratory technique called vitrification, which is a new way to cryopreserve eggs and embryos. And it's really a very efficient technique. And it's allowed us to freeze eggs. Before this technique was developed, we couldn't freeze eggs. It was just impossible. They wouldn't survive. So now we can freeze eggs for cancer patients, for egg donors, and also social egg freezing. And we've recently written a review on this, and I've just got the proofs today. So this will be coming out very soon in a journal called Human Reproduction Update. And this time last year, during International Women's Week, we had, I don't know if you can see that there, but some of us took part in a debate calling, will egg freezing allow women to have it all? And I was for the motion. I said, yes, egg freezing will certainly allow women that can afford to have egg freezing to have it all. And the reason this debate was scheduled was because a number of companies had said that they, as a company perk, they would offer to pay for the egg freezing for their female employees. When I first read that, I felt very uncomfortable about it sort of felt like an exploitation of women offering them egg freezing, but it's an expensive technique. So I think having thought about it and speaking for the motion, I think it is a good perk if the company will pay for it. But let's move on to egg and sperm donor. We will come back to freezing eggs later. So egg and sperm donation has been done certainly since IVF has been around. For egg donation, we need to have the IVF procedure because eggs need to be collected with a minor surgical procedure from the woman, as we would do in IVF. But sperm donation has been done probably for thousands of years, but certainly without sex, it can be done like that as well with various methods. Now, the majority of countries that allow sperm donation have used and egg donation have used anonymous donors. So they felt that it wasn't fair on the donors for their children to be able to trace their biological parent. And all the debates about this, I'm sure you'll be aware that for the children, it's in their best interest to know the identity of their biological parent. But most countries now, some countries now are working on changing that. So the law in the UK changed in 2005. So now any donor used since 2005 in the UK is identifiable. At the age of 18, the child can go to the authority that govern IVF and they can find out identifying information about their donor. So who uses egg and sperm donation? Well, it may be heterosexual couples that can't get pregnant, such as the older age woman or a man with male infertility. We see a increasing number of gay couples. So for a lesbian couple, they'd normally just need some sperm. For a male gay couple, they would need an egg donor and they'd need a surrogate, which I'll come on to in a moment. But also, increasingly, single men and single women who want to have a child. So again, the woman would just need some sperm and the man would need an egg donor and a surrogate. But we can also treat older age women. Now in the UK, this isn't allowed. So this woman here, I think she was about 63. And if you go online and have a look through Google at older age women getting pregnant, you'll see they've been in the news as well. There's many women in their 60s who have got pregnant using an egg donor. They can normally carry the pregnancy themselves. Their uterus stays fine, but they need an egg donor from a young woman to be able to get pregnant. And recently, we've just had a paper accepted called The End of Donor Anonymity, How Genetic Testing is Likely to Drive Anonymous Gamete Donation Out of Business. And there'll be some press release on this once the paper's actually in press, which should be in April. So buying fertility online, or reproduction without sex. What can we do for egg donors? Well, these are real websites. This says these are actual premier donor angels with our program. Beautiful, accomplished, and highly educated. Yes, I think we all feel a little bit uncomfortable with that. There's hundreds of things like this online. We also feel uncomfortable with this. So buying sperm donors. Cryos is the leading, biggest sperm bank in the UK. I was just having a look at their website before I came out, and they said they're in the Guinness Book of Records for having created the most children by donor sperm. I don't know if that's really an accomplishment. So we can just order that sperm and get it delivered to our house and treat ourselves with it. So in the UK, we're regulated by the HFEA if we have treatment at a clinic. If we just buy this sperm and send it to ourselves and treat ourselves with it, we don't go through a clinic. And you may have heard of this gentleman. He was in the press recently. He's claimed to have had fathered 800 children, and he's trying to go for 1,000. He was on TV a few weeks ago. And how he is doing it is this procedure here. So co-parenting is a very bad term. The thing that springs to mind is two people looking after a child after their relationship has broken down, and how they co-parent the child. But that's not what co-parenting normally refers to now. It refers to two people who have met and they have decided they're going to have a child together, and they may or may not, the man may or may not be involved with the child. So you can go to coparents.co.uk. You can also go to coparentsmatch.com, and the people, the women that need one of these donors, these are actually both lesbian couples, but there's hundreds, thousands of them out there. Again, we're looking into this and preparing a paper on the availability, legal, ethical, and social issues surrounding online sperm donation. But yes, this is available online. You don't need to have sex. You can just order your donor online, meet them. Some of the men, most of the men, I think, as far as I'm aware, are doing this for free. They don't charge for their service, and they're bringing a child into the world. So this is legal. It's not illegal in the UK to do this. So now on to surrogacy. I hope you can see this, but this is a surrogacy clinic in India. Surrogacy is where you're renting a womb. So when the original surrogacy was done, it was normally the surrogate's own egg. So she gave her biological child, which she carried for 9 months, to what we call commissioning couple who had infertility. What normally happens now is that the egg will come from an egg donor or the couple themselves. So the couple will often produce the embryo themselves, but the woman can't carry the embryo, so they get a surrogate to do that. But if they do need an egg, they would use an egg donor. These surrogacy clinics in India have been very controversial. You'll see here the women are all lying on the beds. They stay like that for 9 months with very little movement. They don't normally go out and continue with their normal life. They have to stay in the clinic for that time. India have just recently changed the legislation. This was really a huge, huge business in India. There were many, thousands of couples going to India every year to do this. But they've changed the legislation very recently and said you can only do this if you're an Indian resident. And it was I don't think hardly any Indian residents were doing this. It was people from overseas. But you also have legal problems. You have legal problems of getting the baby out of the country it was born in and also into the country that you live in. So there's been a lot of legal cases about this. In Russia, they even have, in some of the clinics, they will take photos of the commissioning woman holding the newborn baby, so it looks like she's just delivered it, and even mock breastfeeding, so that she's got photos to say, no, it was my baby, I delivered it. So I don't know if any of you have read this book, but this is a book by Margaret Atwood many decades ago, she wrote this book, and it's called The Handmaid's Tale. And in this book, there's a situation where rich people, to reproduce, need a young woman who lives in their house and who has their baby, who's their surrogate and an egg donor. And when she has the baby, she gives it over to the couple that she's living with. And that was a really foresight into where we are now. So some of the things that we can buy online regarding surrogacy, view our surrogate angels. And this one, I think you'll all find a little bit disturbing. It says, her body, our baby. I think that just doesn't feel right at all. And that's a real clinic in India. And let's look at how these procedures work with the rich and famous. So Elton John and his husband, David Furnish. They've had 2 children now using egg donation and surrogacy. And you can go online and find many celebrities who have had surrogacy. Some of these women have had children themselves naturally before, but now have opted for surrogacy for whatever reason. I'm not sure whether all of them are medical reasons. I think some of them may be personal reasons that they don't want to carry their own child. So again, the handmaid's tale is very much here. So I'm just going to talk about genetic testing before I bring all this together of where I think the future will take us. So genetic testing is really advanced. I just had a look at the lecture I gave in 2011, and it's just amazing what's happened in the last 5 years. It's totally amazing. So now genetic testing, I'm going to tell you a little bit about genetic testing, a very simple view of it. But we can do it at 4 stages. So if a couple now are trying to have a family, what they can be offered is what we call preconception genetic screening. So they could both do a blood test, or actually they can just use the buccal cells in their mouth, and they can check their genetic code for about 600 genetic diseases, and this will increase with more and more as genetic testing gets refined. They can look for 600 genetic diseases to see if they, as a couple, are at risk of transmitting that to their child. So everyone in here, if we all had our genetic code tested, all of us have some rare mutation. But the risk is, do we meet a partner who also has that rare mutation, which could then lead to a disease in the child? And this has become really popular in the USA, because the insurance companies would rather do this test for a few $100s than accidentally you have a child who then has a genetic condition which they then have to pay for. It's very cost effective to do this. And they're finding that as couples, it's about 2% of couples that they pick up something and then they can then carry on and do the further genetic tests. So if you're found to be carrying a genetic abnormality, we can test the embryo before implantation. That's what I'm going to talk to you about in the next few slides. We can also test you when you're pregnant. So we can do procedures such as amniocentesis or CVS, where we take a bit of the tissue from around the pregnancy and we do our genetic test. I just want to briefly mention something that's really come into the forefront literally in the last 2 years, and this is called non-invasive prenatal testing. So now, from a pregnant woman at around 10 weeks of pregnancy, we can just take a blood sample from her, just from her arm, and we can do something called next-generation sequencing, which I'm going to explain in a minute, and we can find the genetic code of the mother and the genetic code of the fetus. Now, this procedure, I'm sure in the next few years, will be offered to every pregnant woman. At the moment, it costs about £500, and you have to do it privately. And at the moment, we're only looking at the chromosomes, so you can check if there's anything wrong with the chromosomes of the fetus. But soon, we'll do much more in-depth genetic testing and really find out all the information about the fetus. And the beauty of it, we can just do it from the mother's blood. We don't have to put any big needles into the amniotic fluid or into the chorionic villus sampling the placenta to do our genetic test. We just take some sample from the blood. And obviously, after we deliver, postnatally, we can test a baby or any one of us here using a genetic test. Now, I've been working in PGD since 1992, and in 1997, I helped set up something called the STR PGD consortium, and we've done a lot of data on this. We publish data reports every year, lots of guidelines and position statements. And if you want to know anything more about this, you can go to the STR website, and all of the work that we've been doing is freely downloadable from that website. So PGD, or pre-implantation genetic diagnosis, consists of 2 stages. Well, first, the patient has to go through fertility treatment. So even if she's fertile and the man's fertile, we have to generate embryos in our lab. And then there's 2 things we need to do. We need to take some of the cells from the embryo. We can take these little things called polar bodies, which I won't worry you about. We can take one of the cells here on day 3 of development at the cleavage stage, or we can take some cells here, they're called trivectoderm cells, from the blastocyst, which I showed you earlier. Then we're going to do our genetic testing. Now, I just want to explain genetic testing. If we imagine our chromosomes, we've got 23 chromosomes. We've got 22 what we call autosomes, and then we've got the sex chromosomes. So we've got XX for a female and XY for a male. Imagine those chromosomes are like 23 volumes of books. So this is chromosome 1, chromosome 2, chromosome 3. If you open those books, the genes that are contained in the chromosomes are like a chapter in the book. So we'll have a chapter for the gene for cystic fibrosis, for beta thalassemia, etc. And the genetic code is all the letters within that chapter. They don't actually make words, they're just letters. And it's only 4 letters, it's G, T, C, A, that make up our genetic code. So how we do our genetic diagnosis, no matter which stage we're doing it, we can do something called the polymerase chain reaction, or PCR. And PCR is just going to look at a tiny section of one of those chapters. It's just going to look at a segment of a gene, and it's going to copy that thousands and thousands of times. So we have large quantities of that DNA, and we can then do our genetic analysis. So it's almost like molecular photocopying. It's just like taking the one page of the gene we're interested in and copying it thousands of times. So we can then run lots of genetic tests on it and find out whether it's normal or not. Things have moved on since PCR. We do still use PCR, but things have moved on now to DNA chips. So you may have heard people talk about DNA chips before, and there's several types of them, but one that we've been using in our lab is called an array CGH, or array comparative genomic hybridization. And this is where each of our volumes of books are represented by these shaded areas. So this is chromosome 1, 2, 3, up to 22, and X and Y. And these little clones are little areas on the chromosomes. Anything above this green line means there's an extra copy, and anything below it would mean that there was a chromosome missing. So this is actually chromosome 10 that's abnormal, and there's an extra copy of chromosome 10 in this sample, and it's what we call trisomy 10. There's an extra copy of chromosome 10. Now we're at the holy grail of genetic testing. We're at next generation sequencing, or NGS. So this is where we can use it at the moment very easily just to look at the chromosomes, but over the next few years we're going to go much more in depth and really be able to read the genetic code in much more detail. And it was James Watson back only in 2007, and it cost $1.5 million to sequence his genome, and it took many, many months. I couldn't find out how long it took, but it did. This is a page from the Illumina website, one of the companies that do this. This was taken a few years ago, and it says here it cost $9,500. And this was a more recent screenshot, someone else offering sequencing. One of my friends at Illumina told me just now it will now take 24 hours to sequence my genome, and it will cost $1,000. He's trying to get Illumina to do it for free, because I'm like a guinea pig for them to be able to talk about it. So for $1,000, you can have your genome sequenced. So I was a guinea pig recently, to the horror of some of my colleagues. There's a test that you can buy in Superdrug. It costs £125, or you can buy it online. I just bought it online. And it's called 23andMe. It's not sequencing yet. It's using one of these DNA chips that I told you about. But this is me in my lab with my sample. So I had to spit into the tube so that some DNA went in. And then I got my results. And I've got pages and pages of results. I've been on Radio 4 a couple of times talking about the results and how complicated it all is for any layperson to really understand. But here you can see cystic fibrosis, beta thalassemia, and some other common genetic diseases. But where is the very last stage of this? And the very last stage is actually genome editing. So I've talked about checking our genetic code, but what about editing our code? So you may have heard a lot about this recently, CRISPR-Cas9. And this is a very cheap and easy way to precisely edit the genes. So we can develop gene therapy to treat diseases. Recently, there's a report of some stem cells that were containing a gene that caused blindness, and they edited the gene in the stem cells and corrected it. So we could prevent genetic disease, we could edit genetically abnormal embryos, we could introduce, enhance, or eliminate traits for non-medical reasons. But anything we do would be passed on through generations. Once we've edited that genetic code, it would be passed on. So we don't know what that's going to do. This is really a genetically modified person. And I think probably most people feel a little uncomfortable about genetically modified food. Well, this is genetically modified humans. We don't know what it's going to do to our ecosystem or our planet. So it's a real unknown. And the reason why it was in the press in the UK a lot is because some colleagues at the CRIC were trying to get a license to modify human embryos. It has already been done in other countries, such as China. They've already been working on this. But this license was approved by the HFEA. And I recently took part in a debate at a genetics conference that was called, Should We Be Using CRISPR-Cas9 to Experiment on Human Embryos? So the future technologies, let's bring all of those things together. Will we actually need egg and sperm donors? Will we need egg freezing? Will we need surrogates? Will we need to do PGD? Well, maybe not. There are people that are studying making artificial eggs and sperm from stem cells. So if that works, if we could take some of my skin and make some stem cells and then zap it and make me a nice young egg, I wouldn't need to freeze my eggs. I could just make a baby with my eggs whenever I wanted to. We could make sperm from stem cells as well. So we wouldn't need to worry about egg or sperm donation. But also, we maybe don't need to worry about egg or sperm donation because we could just clone ourselves. Again, we could take some of my skin cells, make some stem cells, and clone myself. I could even carry my identical, genetically identical twin or individual, whatever you like to call it, clone. So maybe that will totally outweigh the use of donor eggs and sperm. And artificial womb, there's been a lot of discussion about this recently. Many people are working on trying to develop an artificial womb. And there was a goat a few years ago that was grown in an artificial womb. So maybe we don't have to worry about surrogacy. We'll overcome the need for a surrogate because we'll have an artificial womb. Maybe we'll be doing PGD for everything. At the moment in the UK, we can only do PGD for the genetic disease the couple carry. But if we do in-depth next-generation sequencing, we'll be able to find out about the full genetic code of that embryo. So will people decide, actually, I won't reproduce the old-fashioned way. I'm going to reproduce without sex. We'll make some embryos, and from my 10 embryos that I generate, I'm going to pick the one that is genetically most suitable for me. So maybe both the parents are concert pianists, and they want to be sure that the child they're going to have is going to have the best set of genes to give them the best chance to have a child that's got musical ability. So using PGD, we're not changing anything. We'd have to just take the embryos they produce. They might not produce one that's got the best genes to be another concert pianist, but they could certainly select that, and that will be possible very soon. The talk I gave in 2011, at the end I said, hopefully in the future not everyone will be reproducing by PGD. And in 5 years, I'm saying, well, I think probably we're there. There are clinics already that are offering PGD for every patient. But maybe we don't have to worry about any of that. Maybe we just produce one embryo and then we edit it to make it what we want. So if we want the child that's got actually not just the best pianist, but also the sportiest, the tallest, and everything else will make this perfect child. I just hope they live up to our expectations. It's not all genetics, but we could edit it to give it a predisposition to be that. So one problem we have is who's going to go through this treatment. It's only going to be people who have got the finances to do this. These techniques do not come cheap. PGD certainly costs about £10,000 for one cycle. CRISPR is a cheap technique, but you'd still have to go through IVF to do it. So Gattaca, if you want to Google Gattaca, for those that haven't seen it, though some of you have seen it, it says in this movie, in the near future, everyone will reproduce by PGD. And I'm afraid that we're almost there. There are clinics, as I said, in the US that do PGD on every single embryo. They're not looking at the moment for genes, they're just looking at chromosomes, but that's only a matter of years away. And then who will go through CRISPR? Who will have the finances to go through this? And we'll end up with reproductive and genetic classism. So in conclusion, IVF treatments are going to continue to be developed. IVF is a multi-million-dollar business. My children were born by IVF. I've got a son after 4 years of infertility treatment, finally gave birth to my son just before my 40th birthday, and I have IVF twins that were born from frozen embryo transfer. So I know what it's like to go through this, but it is going to just be increased, especially as women get older. It's a huge commercial market, and people are going to cross borders and buy fertility on the internet and make new types of families, as we're already seeing. So all of this is going to involve reproduction without sex. There'll be an increase in personal genetic testing, and I'm pretty sure that probably in the next 10 years, certainly in my lifetime, everyone's going to know their genetic code. It makes sense as far as the health service as well. If the treatment that you have and how you're actually managed will depend on your genetic code. We could help predict what lifestyle changes people would have to make to stop them getting some disorders. So it's very sensible to know our genetic code. as long as we understand everything about genetics, which we don't do at the moment. So we're going to see more PGD, we're going to see more gene editing, and I certainly feel embryos are going to be enhanced. At some point, we're going to use this technology. Certainly that's not the initial aim at the moment, but everyone wants to have technology. Everyone wants the new phone, the new car, the new TV, and it's going to continue in what we're doing here as well. So this is a quote from Edward Tatum. It said, "The time has come, it may be said, to dream of many things, of genes and life and human cells, of medicine and kings." And just bringing all that together, we've got our super eggs and sperm for our egg donation. We've got our frozen embryos. We've got surrogacy. We've got our genetic testing. And Aldous Huxley, I think we should all reread this book, Brave New World. It was certainly a time where everything I've said has come together. And when I read it many years ago, I didn't think it would be happening in my lifetime. But it's here. It's certainly here now. And we discussed the ethics of what we do. A number of us have got together at various times. We published this paper a few years ago, and we're having another meeting of the groups of the European Society of Human Genetics and ESHRA coming up in September to discuss all these issues that I've spoken about. And this is some of our team that have contributed to this work over the years. Some of these people have left now, but they should all be acknowledged. And just to say that this talk is part of the International Women's Day events, and in the Institute for Women's Health, we've got a great program of events. We had Rights last night, which I went to, about female genital mutilation, which was very emotional play. And this afternoon, in this room, at I think it's 6 to 6:30, we're having a debate on buying fertility on the internet. So if you've enjoyed my talk today, please come along to that. And there's other events here that are happening over the next few days. And I'm very passionate about education. So last year I set up a public engagement group called Global Women Connected. You can find us at globalwomenconnected.com. And we talk about many issues. We're trying to make sure that people become educated about some of the things I've talked about, but even broader women's health issues. Thank you very much.
Thank you very much, Joyce. So you won't need drugs to win at tennis or athletics or anything.
No.
You just need to have been designed properly in the first place.
Yes.
So I'm opening it for questions then. There's one here for the first one. Hang on for the mic.
I know the technology is early. It's 38 years experience. In fact, I live very nearby the first baby that was born by this technique. My question is there any evidence yet, significant evidence of a difference in morbidity between children born by these techniques as opposed to the old-fashioned technique?
The fun way. Yes, this is a question that's put a lot of the time. So Louise Brown, obviously she was born in 78, so she's not very old at the moment. Every now and then someone will publish a paper saying something terrible is going to happen to these children. It is quite invasive. We give the woman fertility drugs, we culture embryos out of the body, we're doing all sorts of things. It's something that I personally have been aware of, and years ago I had a long debate with Bob Edwards about, what are we doing? Are we pushing things back and back and with all this new technology? So we just don't know. We are following the children and seeing what happens. You will see a paper that says, they have different neurological profiles to normally conceived children, and they have this and that and the other. But then you'll see another study that says, no, our study's not showing this. So we are following them. We just don't know. We hope everything's going to be okay. But for these couples, the option is no children. And most people want children. So we're using them. But we have to be careful. Yeah, absolutely for sure. Reproductive cloning produces very abnormal offspring. Almost every species has been cloned. And they're really quite abnormal. So we have to wait and see. Is there another question? One over there. Yes.
So me and my girlfriend have been talking about having babies, but obviously we can't have babies. What you were talking about with taking DNA and possibly turning it into an embryo or a sperm cell or whatever it might be, do you think we're quite close to possibly doing that?
Do you know, so one thing in all of this, in all of the new technologies, not just in my field, but in other fields, we just don't know what's going to work. So, for example, I remember in the early 90s, I was told that gene therapy was round the corner. It was going to work, it was going to be amazing. And, you know, we're in 2016, and there's very few gene therapies that work. You know, about 15 years ago, stem cells were it. Stem cells were going to cure everything, and it was going to be amazing. And we're still not there. Now it's CRISPR. Is it going to cure everything? So medicine sometimes goes a lot slower than we anticipate. The non-invasive prenatal testing I told you about, it had been going on, the research had been going on for over 20 years, and nothing had happened. And literally overnight, the next-generation sequencing worked, and it has revolutionized pregnancy testing. It really, really has. And it was one of the, I think it's the fastest technology that was ever introduced into the clinic. And it's going to revolutionize testing when you're pregnant. And so we just don't know. And I've asked my students, only from the last couple of years, that I want them to predict what they think will be the technology that really takes off in 10 years' time, because we just don't know. So we don't know. So I wouldn't risk thinking, oh, it's okay, when I'm 40, we'll be able to use some of my skin cells to make an egg. I just don't know. I'd always said to my students, 1 year I'm going to tell you that you should freeze your eggs. And my master's course, my main master's course started 20 years ago, and it's only been the last 2 years I've said to them, I'd freeze your eggs if I was you. You know, it's taken all those years to be able to say that. So we just don't know. Very unpredictable.
I'm sorry, I've got to end it there. It's time to finish. So much to think about.
Yes.
Ever so. Big hand for Joyce.